Hello, everyone, and welcome to today's webinar, Sorting It Out, a comprehensive webinar on single-cell flow cytometry. I am Morgan Sterling of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of our presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Angie Goldfain, Senior Product Manager at Thermo Fisher Scientific. Angie, you may now begin your presentation.
Thank you for the introduction, and thank you all for joining today. Flow cytometry and cell sorting are growing areas in research. My hope with this seminar is to provide a basic overview of flow cytometry and some key areas to consider when adapting this technology to your single-cell research. I'll focus on a high-level overview of flow cytometry and cell sorting. We'll talk a bit about instrumentation and setup, sample preparation, and then some basic cell sorting applications to which this technology can be applied. While there are several methods available using magnetic beads and microfluidics, for the purposes of this talk and in the interest of time, I'll focus on cell sorting using the electrostatic droplet method. Flow cytometry is the measurement of cell properties while flowing in a single cell suspension. And that single cell suspension is really key. Some of you may have used a cell analyzer or cell sorter before. A cell analyzer collects data from each cell for analysis only, so it provides you the data, whereas a cell sorter does that and takes it to the next level and lets you separate the cells of interest based on that analysis. As you can see in the diagram to the right, this really starts with a single cell suspension, which is focused into a stream and then interrogated by one or more laser beams. Single cell suspension is in a carrier fluid, which is called the sheath. This presents the cell to that interrogation point, and that's where the lasers hit the cells in the stream of fluid passing by. From there, we collect light coming from each cell, separate by wavelength, and digitize for processing and display to the user. This is where the analyzer ends and the sorting picks up. For an electrostatic droplet sorter, we use a crystal in the nozzle, which vibrates the stream and causes droplets to break off. If a population is identified for sorting, the system waits until that cell is in the last attached droplet, where it is charged positively or negatively, and then moves through an electric field and is deflected into the collection vessel below. This can be test tubes, microtiter plates, or other media. I suggest you always use the suggested QC procedures to ensure your sorter and analyzer are working properly before running your samples. So what happens when the laser beams hit the cells in the stream? A key aspect of flow cytometry is the generation and collection of light. As each cell or particle passes through the laser beam or beams, it causes the light to emit in all directions. There are really two types of light that we look at. One is scattered, and you'll see that on the left-hand side of this slide. Anything passing through the laser beam scatters light. So any particles, background, debris, or the cells we're looking for. We look at scattered light in two ways, both in the forward direction and the side direction. The forward direction gives us an indication of relative size of the cell, whereas the side direction gives us some information on the internal complexity. Some systems exist where you can see more than one wavelength of light with the scatter as well, if you're looking at small particles or alternate wavelengths. Now, the fluorescence is a whole nother side of the light we look at, and that is demonstrated in the image on the right. What happens is the laser light excites fluorophores, or any tags that you've added to your cell suspension, absorbs that light, and then emits in a longer wavelength. That's called a Stokes shift. It's longer wavelength. And then we collect that light, that longer light, and we can tell what fluorescent molecules were on the surface of the cell when they were excited. We can mark internal and external components of cells. So how do we label cells? Well, again, as we come back to the single cell suspension, it's really important, and that's key. And by using that, then we add antigens, antibodies, or different dyes and stains to mark the cells of interest. This can also be fluorescent proteins within the cell already, or autofluorescence of cells. Now, an antibody added to the cell suspension can be tagged with what we call a fluorophore. This is what absorbs the laser light and then emits the fluorescent light. Antigen abundance and fluorophore selection are really important things to consider when designing your experiment. Not only does it need to align with the instrument you're using and the lasers available, but also focused with the cell type that you're interested in. There are viability dyes as well, and this is really important to make sure you're only considering the live cells and eliminating the dead cells if you're sorting for that purpose. We can also look at DNA content as well, or like I mentioned, the fluorescent proteins. Many of these options can be used on the same cell, so therefore cells can emit many, many different wavelengths. And then those have to be separated to identify the populations of interest. So how do we separate that light? We use optical filters. Optical filters are just coated pieces of glass that can separate the light into the different wavelengths and steer it to the detectors. So we have 4 main types of filters in flow cytometry. There are a few others. These are the 4 ones you'll hear most. A dichroic long pass, which lets some light pass through and reflects others. A dichroic short pass, which lets the shorter light pass through and reflects the others. A band pass, which lets a specific band of light come through. And then neutral density, which just generally dims everything down, kind of like sunglasses for that signal. The same process of filtering and bouncing light is used for both scattered and fluorescent light sources. And these filters direct the light directly to the detectors for amplification and processing of that light. So what happens once the light's been separated? I said it goes to the detectors, which can be PMTs, photomultiplier tubes, photodiodes, or other types of up-and-coming detectors. What those detectors do is amplify the light and send it to the electronics for processing. So the electronics take that signal coming from the detector and process in several different ways. We, first of all, have to set a threshold. And this threshold level, as you can see in the diagram on the right, shows the level that a particle needs to exceed in order for it to be important. So we're trying to eliminate any baseline or background noise or particles or debris from the assessment. Once it passes that threshold, we measure other properties of the cell or the pulse. The height, how high it is, the intensity, the area under the curve of that cell pulse, and the width of it. These measurements give us a lot of information about every single cell, both the scattered and fluorescent light. For example, in this case, if we were to look at it, the scattered light would give us an idea of cell shape again, or size again, and the side scatter shape. A large fluorescent pulse may point to abundance of that marker on the cell surface, or perhaps the protein expression. So it's very much relative to other particles that are in the suspension. This process takes place for every detector on every cell that crosses that threshold. So a system with, say, 7 lasers and 50 detectors would provide over 1,000 different measurements for each cell that passes through the laser beam. And we can run at different event rates. You can run at hundreds, thousands, even tens of thousands events per second, so cells per second. So the amount of assessment we're doing on every cell is massive. So how do you boil that down to actually see what you want? This is where data visualization comes in. So every cell sorter comes with a computer and software package which displays the data to the user for analysis and assessment. These files are called FCS files, flow cytometry standards, and there are a variety of offline software packages available which give you the ability to read and process these files for advanced features. Basically, the data is displayed in plots, histograms and density plots, some of which are shown here on the screen. In these plots, the user can select which parameter they'd like to see on each axis and which measurement, the pulse, the width, the area, etc. Regions and gates are used, which you can see again in these diagrams, ellipses, polygons, bar regions, to identify the populations of interest and gather more information. I could spend multiple webinars just on the different aspects of data visualization, but that's not the purpose here. So suffice it to say, there's a lot to identifying which cell populations are desired for sorting, including running proper controls and compensating for overlapping spectra. Once you know what you want to sort, you can create regions and decide on the experimental needs for purity and recovery, among other things. So now that you've identified what to sort, how do you know it will be sorted properly? The key to this is timing, or one of the keys. That timing is called drop delay. Drop delay is the timing between when the lasers hit the cell of interest and that cell moves down the stream to the last attached droplet. As we talked about, remember, there's a crystal in the nozzle that vibrates, causing those droplets to break off at a very steady rate. So getting that timing right is really key to sorting the right cell at the right time. There are different methods for this. One of those is using beads, which are small plastic particles, and adjusting the timing while watching for sorted events. That's the diagram you can see on the right here. On the x-axis, we have drop delay, timing, and on the y-axis, intensity of what we're seeing. We adjust the timing and we look for the intensity of the events to increase. This means we're nearing the optimal drop delay. We continue past that to make sure we found the true peak and therefore the most accurate drop delay timing setting we can. This is so critical, as if you're off by just half a droplet, it can give you no cells back or very minimal, or the wrong cells, maybe even worse. So this is really, really a key part for cell sorting this way. So how do we sort from there? Once we've calculated that timing, we have to know which direction you're going to sort and how. So what we do is we apply a charge to that last attached drop that we've just accurately timed. We let that droplet break off from the rest of the stream, and then that droplet goes through an electrostatic plates for deflection. We call them deflection plates. So the negatively charged cells will move towards the positive and the positive towards the negative. And then it directs them into whatever sort collection vessel we have below. We also catch the waste, which can be decontaminated safely in the future. So different instruments can sort at different speeds, and different number of populations into different collection vessels. Really, the world is your oyster with this. One of the key factors is the speed of droplet creation. Some instruments you can create 30,000 droplets a second, some 90,000 or 100,000 droplets a second. So this will really dictate how fast and how quickly you can sort the cells. We talked earlier that you can run hundreds, thousands, or even tens of thousands of cells per second. So that combined with your droplets per second will give you your limiting sort rate. It's also highly dependent on the electronics for the instrument. Again, if we're measuring over 1,000 things per cell and we're running at 1,000 events per second, for example, you've got a lot of data coming through there and a lot of decisions to make really quickly in order to sort the right thing. So you have to ensure your electronics can keep up with the sort rate prescribed for your experiment. Some instruments can sort different ways, 2, 4, even 6 ways into 5 ml test tubes. There are other collection vessels as well. Some people sort into microtiter plates directly, and this is a key for single cell sorting. You can sort into 96 or 384 well plates very accurately and have exactly 1 cell per well, which you can do further downstream processing. You can sort into slides, into Petri dishes, PCR strips, anything that you can think of and put underneath there, we can sort into downstream processing. So once you've set the timing properly, how do you know what's going to get sorted and what's not? In all cell sorting experiments, there's a balance between the purity of the sorted sample and the quantity of cells recovered. Sort modes allow us to adjust this balance. There's an enrichment sort mode, and these sort modes are pretty consistent from instrument to instrument. They'll have very similar modes. An enrichment mode sorts everything that's positive, everything that's past your reagents and gates, even if there's something negative present. This means you'll get every cell back that you want, so your recovery will be high, but your purity may suffer. And from the diagram on the right, you'll see the enrichment, all the droplets labeled with the green arrow would sort. Therefore, you will have some contamination. Now in purity mode, that's the most commonly used. It sorts everything positive as long as there's nothing negative there. So again, in the diagram to the right, you'll see those are labeled by the purple arrows. So that would sort those 3 droplets and not sort the ones with the contaminating events. This keeps recovery reasonably high, but really improves your purity as you're not planning to sort anything negative. Another mode we have is single mode. Single mode is meant for when you want to sort one and only one cell, and you don't even want multiple positive cells in a droplet. This is when you're commonly used for cloning and sequencing applications, where the placement of that one and only one cell is critical to any downstream processing. Your purity is really high for this. You're getting exactly what you want, but your recovery can be low, so you could be throwing away some cells that you want. Some systems have additional options where you can adjust the window used to make that sorting decision, the droplet envelope, the number of drops we're sorting, or even collect aborts in a different tube. That's called mixed mode on a lot of instruments, where we can sort at a high purity in one direction and collect the aborts in another direction. This allows you to have a really good balance between both purity and recovery. Let's switch gears a little bit and talk about sample preparation. This is really key to cell sorting. No matter the instrument you're using for sorting, there are a number of basic guidelines, so some of which are listed here. Let's go through those. As we talked before, single cell suspension is really, really important. Remember, we're pushing the sample through a nozzle or a small orifice, depending on the system, and if we have clumps of cells, that will clog that orifice. Clogging is something that happens with cell sorting, but we'd like to avoid it as much as possible. So as much as you can do to get a single cell suspension, the better. We also always recommend filtering samples to remove any clumps or residual particles from the prep. So that's always important too, again, just to maintain that single cell suspension. Consider cell size and system setup when you're prepping your samples. Think about your nozzle size, think about your pressure. Do you have really fragile cells that you're sorting that are very sensitive to pressure? Do you have more flexible cells and they're fixed so you can go faster and you can treat them a little more harshly? Keep that in mind, very important. And with panel development, again, that's a whole nother area for discussion. I'm sure many of you have seen webinars on that as well, but that's an important element to this as well, like the complexity of the panel that you're using on your cell sorter. Many cell analyzers can look at more information around a cell, and when you get to a sort, you want to try to trim that back to the critical features. When you're also designing a panel, you want to consider titrating your antibodies. You have to identify which amount of antibody is needed to give you the best separation between your positive and negative populations. In the little histogram on the lower right of this screen, you can see a titration experiment, and each of those showing different concentrations of antibody added, so you can find the maximum separation between the negative and positive cells. I always recommend including a viability dye if you're working with live cells. Like I mentioned previously, it's really key to ensuring that you're getting the cells you want, and those are alive if that's what you're looking for. Always, you need controls. We're all scientists, and for any experiment, you need controls. So make sure you have the proper controls for any analysis or sorting experiment. This can include single color controls to ensure your staining and prep went properly, negative control to look at background autofluorescence, and even fluorescence -1 controls if you have any concerns of overlapping populations. All of these are really critical to ensuring the quality of your experiment. Another thing to do is to consider temperature control. Really, you got to keep the cells happy through the cell sorting process. This is really key. A lot of instruments have temperature control both on the sample input side and the sort output side. Keeping the cells at a steady temperature throughout the sorting process will really help with your overall viability and recovery. If your cells like to be cold, 4 degrees, if they like to be warmer, you can adjust that, but keeping those cells happy. Consider this, cell sorting. We're putting a tube of single cells on an instrument. We're jetting them out through a tiny little orifice. We're hitting them with laser beams. We're shaking them to create droplets, and then we're charging those droplets and deflecting into tubes. This is putting those cells through a lot, so whatever you can do to keep them happy. And one of those things is to give them some media for cell collection. Give them a swimming pool to land in. That's what I like to say. After going through all that, give them something. Always practice safe sorting. With cell sorting, in this method at least, we are creating droplets, which are aerosols, which are a good size for any person to breathe in. Aerosols can be biohazardous depending on the cell type you're running. Of course, aerosolizing anything makes it more dangerous. So always consider safe sorting, consider aerosol containment methods, aerosol evacuation systems, even biocontainment enclosures to ensure you're keeping the people safe that are doing these experiments. It's also important to keep the product safe, and so that's something you might consider as well, to make sure you don't contaminate what you're actually sorting. Some of that can be passive by just having doors in place, and some is the more aggressive airflow just to maintain that sterility inside that sample chamber. I recommend checking ISAC guidelines. ISAC is the International Society for Analytical Cytology, and they have good guidelines for biosafety based on the type of cell that you're sorting. So let's talk a little bit about cell sorting applications. Cell sorting generates live homogeneous cell populations. You can do those by bulk into tubes, or like we've said, into microtiter plates, a single cell, for downstream applications. Isolating these pure populations really can enable you to make new discoveries, and this can happen quickly too. You need to consider all the different aspects of the cell type you're sorting so you can decide on the method of sorting to apply. Some of these applications are listed on the right, from genome editing to cell therapy, protein expression studies, and more. There are even more not listed here. We have people doing chromosome analysis, marine biology studies of phytoplankton, and even identification of extracellular vesicles. So really the sky is the limit when it comes to the applications for flow cytometry. You have to know your cell, know how it likes to be treated, and know what you're looking for before starting any flow cytometry experiment. Here we have a few additional flow cytometry learning resources for you. Thermo Fisher is really focused on education, and we want to make sure you have every bit of information you need to design a panel well, to have the tools you need to sort or analyze, to create your protocol, and identify the reagents needed. We even have a space for spectral flow fundamentals. So if you're just getting into flow cytometry, spectral flow cytometry is a whole new area that the science is moving towards. And we have overview and educational seminars on that. So thank you so much for your time today. I appreciate it. And I think we'll move to questions.
Thank you, Angie, for your informative presentation. Before we jump into our live Q&A, we do have some questions for our audience. First one is, and we'll give you a minute to answer these. How many of you have used or are currently using a cell sorter? First answer is, I've never used a cell sorter and I'm looking forward to learning how to get started. Second option is, I've used a cell sorter in the past, but it's been a while, or I relied on the help of an expert. Or C, I regularly use a cell sorter in my research. We'll give you guys a minute to answer this. Take your time. Awesome. And we'll move on to our next poll question, which is what technical feature is most important to you when choosing a cell sorter? Number one is number of fluorescent channels. Number two, speed and or high throughout plate sorting capabilities. Or three, biosafety capabilities. We'll give you a moment to answer. All right, and our final poll question before we move on to our live Q&A is how important is spectral functionality on a flow cytometry based cell sorter? Very important, somewhat important, but could do without, or not a priority for me. Again, we'll give you a moment to respond. Great. Now let's jump into our live Q&A. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll go ahead and answer as many questions as we have time for. So let's get started on our first question, Angie. What is the size range for cells using this type of cell sorter?
That's a great question. And it varies based on the instrument you're using and the nozzle tip size. That's probably the most critical factor in this. You want to make sure that a cell is about 3 to 5, or nozzle tip's about 3 to 5 times the size of your cell when you're sorting. And that's so that you don't get a clog, right? You don't want a couple cells sticking together and clogging that nozzle tip. So some instruments have nozzle tips ranging from 70 microns up to 150, and you would adjust the nozzle tip based on the size. Now, if you're talking about size range of cells, it depends. Instruments can go down to 0.2 microns or even smaller. If you're able to fluorescently label a cell, then we can detect even smaller than that. And some instruments actually have specific small particle detectors for this very purpose. And that usually uses the 405 nanometer laser, and we can see those smaller particles and sort them. So if we can see it, we can sort it, right? So if you can either see it via scatter or fluorescence, we can separate it. So the practical range is maybe 0.2 microns up to somewhere around 40 microns, depending on the nozzle tip size you use and the instrument capabilities.
Awesome, thanks. Next question. Does drop delay stay relatively the same between events, even if the cell population is heterogeneous?
So when you're calculating drop delay, different instruments do it differently. In this case, the image I showed came from the Bigfoot cell sorter, and we actually calculate the drop delay based on the middle of the pulse. So in that way, since we're looking at the very center of the pulse coming through for drop delay, it doesn't matter if that pulse width changes based on cell size. So the drop delay does stay consistent despite the different cell types you might be sorting. Some instruments do look at the beginning of the pulse, and in that case, the drop delay timing might change slightly as you change sizes. So I always recommend doing a quick test. You can sort onto a slide usually just to verify with cells, cell-based verification of your drop delay with those actual cells that you're sorting to ensure that you have an accurate timing set.
Great. Next question. How do you know what fluorophores to choose for your cell sorting experiment?
It's dependent on several things. First of all, of course, you need to know your cells and what you're looking for and what antibody fluorophore combinations are available. So that takes some time to develop, of course, based on your application. Fluorophores need to be catered towards whatever instrument you're using as well, right? You don't want to use a fluorophore that is excited by UV if the instrument you're going to use doesn't have a UV laser. So it's very important to work with whoever the manager is of that cell sorter that you're going to use to make sure that the fluorophores you're selecting will match the configuration and can be used. I would also, though, highly recommend reaching out to a panel development specialist. Most companies actually have them. I know at Thermo Fisher we do, and they can really help you optimize your panel development, fluorophores, and how much they overlap based on the different populations you're looking for. So I always recommend going to those experts for some advice, especially as you're building bigger and larger panels and you're going into that spectral space. That's really important.
Awesome. What are the most important factors in keeping cells healthy through the sorting process? What if my cells are really fragile?
So there's a lot you need to keep in mind when you're sorting cells. First of all, think about the temperature that they like, the temperature they're coming from, the temperature they're going to, and you want to make sure and set up your instrument to accommodate that, right? The second thing is you'd want to definitely filter your samples, start out so they're not too clumpy at the beginning, and make sure you add some media or maybe FCS, something that can keep the cells happy, some sera, different things you can add to make sure the cells aren't sticking together and have what they need. Make sure you sort into the media they like as well, whatever that might be, whether it's culture media, whatever the cells are going to like, and keep them healthy. A few other factors to keep in mind is different cell sorters use different types of nozzle sizes, right? As I mentioned earlier. And you want to make sure if you've got a really fragile cell, you might want to go with a slightly larger nozzle tip size so that the shear forces on that cell are a little bit less. A lot of instruments also allow you to adjust the pressure on the system, and this can be really key for fragile cells as well. You can treat the cells more gently if you run at a lower pressure with a larger nozzle tip, and thus increase your viability after sorting.
And how effective are these with respect to viruses, or what is the relationship with viruses using these?
So viruses, yeah, those are pretty small. So it depends if you can fluorescently label particles or the viruses for sorting. I'm not sure I understand exactly what you mean by how effective these are, but I think as long anything you can label fluorescently, we can sort. We have some people that sort chromosomes, for example, or other small particles, and basically, as long as you're able to fluorescently label them and you can see them, then that's something we could separate. So that would be a key to sorting really anything.
Awesome. And we have an example here. I have identified a cell population on Aurora, a full spectrum cytometry. They're wondering if Bigfoot could have similar fluorochrome sensitivity as Aurora.
That's a good question, and it's something we certainly get asked a lot. As Bigfoot is a full spectrum sorter, and Aurora is, or the current Aurora, there is a sorter available. The most, I think what you're referring to is the analyzer, is a little bit different. So they do have different optical setups, so you will see differences. You'll see differences between any two instruments you switch to, just due to the differences in laser power, optical filters, detector sensitivity, and almost as important, either jet and air versus cuvette based. So the Aurora is a cuvette based analyzer, right? And so you get a little bit more separation generally and sensitivity than you do in a sorter with jet and air. So that will vary a little bit. Now, that doesn't mean you can't use a sorter after you have a good panel established. It just might take a few modifications, which is why one of the reasons we have a panel development team at Thermo Fisher, because we want to make sure and work with you if you do have an Aurora panel. How easily can that transition to the Bigfoot, or what modifications might be made to make that even better? So generally, going from an analyzer to a cell sorter, you'll lose a little bit of sensitivity, but you can generally compensate for that as well with optimizing your panel design.
Awesome. And Angie, what steps do you recommend to get clumpy cell types into a suspension that won't clog?
That's always a fun question, right? I always recommend filtering samples before you run them. That's always a critical thing. Get rid of any clumps in advance before they go through the sample line and the nozzle tip. And there's other things you can do. There are chemicals such as pluronic and other things like that, which actually you can add to your sample to make the cells a little bit more slippery, for lack of a better term, or less likely to clump. And so that actually could help quite a bit if you're concerned about that. I also recommend usually keeping the cells cooler, generally a colder temperature. The cells won't clump nearly as much as if you keep them warmer. So temperature can be a big thing, and the media that you have them in. And then finally, I always recommend an instrument with some agitation while you're running, so that if you can mix and agitate that sample as you're running and acquiring it, that will help the cells stay in suspension and help prevent them from clumping.
Awesome. Looks like we have one more question, so we'll end with this. What safety precautions do you recommend for human blood?
So I think you always should, first of all, consult your local safety officer based on the samples you're running, right? And the potential for the severity of whatever that sample can contain. However, I always recommend some sort of aerosol containment or biosafety enclosure for really any live sample. Because if you think about it, anything really, we are making droplets. So we're enclosing cells in droplets that are a great size to go right into your lungs and potentially cause illness. So I always recommend having some biosafety in place for any live cell sorting. And I know that Isaac recommends the same. Now, it depends, of course, on the risk factors. And so, like I said, always consult your safety officer and make sure as well that you clean everything thoroughly after, and you use universal precautions for handling, right? In terms of wearing gloves as you're taking samples in and out, making sure they're capped before they're removed to the room, letting that aerosol evacuation system run for a little bit of time after your sort completes, just to clear any residual aerosols in that chamber. So I think there's a lot of things that can be done if you're running samples that you're not sure what they contain. But always get that information first and work with your safety officer so they can establish clear protocols on what you're doing. And like I said earlier, I mean, safe sorting is really important. It's something that Isaac has been stressing lately. Really want to reduce any laboratory acquired infections.
Great. Well, thank you so much again, Angie, for your time today and your important research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast will be viewed on demand. Labroots will alert you via email when it's available for replay. We do encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you so much. Take care, everyone. Goodbye.